Good afternoon. We actually have an exciting afternoon with Walter Greenleaf, David Axelrod, Mary Lou Jepson, and Vinod Khosla, and beginning with Ed Boyden. Ed Boyden is a professor of biological engineering and brain and cognitive sciences at the MIT Media Lab and the McGovern Institute for Brain Research at MIT. He leads the Media Lab's synthetic neurobiology group, which develops tools for analyzing and repairing complex biological systems such as the brain, and applies them systematically to reveal ground truth principles of biological function and to repair these systems. Welcome, Ed Boyden, but where are you? Oh, okay.
All right. Great. All right, it's great to be here. So I direct a group at MIT that works on technologies to try to understand and repair the brain. And if I had to pick an overarching goal, I would say our goal is to really understand the brain to the point where you could make a biologically realistic but comprehensible computational model of how a brain circuit works. And as a byproduct of this quest, of course, we want to help treat diseases, maybe even use the C word, cure, someday. But for now, what we want to do is to really gain an understanding. Although I hope I'll show you throughout the talk that as a byproduct of this understanding, we are spinning out companies and helping get technologies out into the world. So 3 core goals that we have include mapping the molecules and circuitry of the brain, controlling the high-speed dynamics of the brain, precisely and ideally non-invasively, and then finally watching the high-speed operation of the brain. And I would argue that this has been difficult because, of course, the brain, unlike many other biological systems, certainly not all, uniquely has to span so many different scales of time and space. So, of course, brain cells are amongst the biggest cells in the body. If you count the spinal cord, of course, they are the biggest cells in the body. And yet, if you care about neural connections, if you care about receptors and ion channels and all the other molecules that make neurons do what they do, then you have to get down to nanoscale things. And that, of course, presents a great tension. And of course, the other issue is time. So if you care about Alzheimer's disease or Parkinson's or cancer or of the brain or other things, then you have to deal with very long timescale events. But the briefest events in the brain, these millisecond timescale electrical pulses within neurons and the chemical exchanges between them are incredibly brief, right? So how do you cross those temporal domains? So we really want to cross time and space, and to do that, we've been developing 3 tool sets to help us achieve that. And I'll tell you 3 short stories today, the last of which was only published this afternoon, where we are trying to bring forth new technologies to help us really map, control, and watch what's happening in the brain. So let's jump right in. Let's start with mapping. So mapping the brain, of course, is a pretty big enterprise. Many people have been trying to map it non-invasively with MRI and other related methods. Later you'll hear from Mary Lou about her really cool stuff. You also hear people mapping with electrons, tiny, tiny regions of the brain, but of course it's difficult to get molecular information. And then finally, super resolution methods, of course, have gotten a lot of attention. Some of the inventors won the Nobel Prize a couple years ago for their efforts. But you get molecular information, but you don't go very fast, and it's hard to image large 3D objects. So we thought, what if we do the opposite of what some of these attempts have been like and try to blow everything up? And so starting with 2 really great grad students at the time, Fei Chen and Paul Tilburg, we decided, what if you could physically expand brains? So obviously this is not going to be done on a living brain. It would be done on a preserved piece of tissue. But I hope to show you in the coming slides that there's still a lot that we could learn about the normal and pathological brain by mapping the molecules throughout cells and the cells throughout circuits in this way. So how does this work? Well, we want to install a swellable polymer evenly throughout a preserved piece of biological tissue. And so this owes debts to 2 old pieces of work. One is from the physicist Toyoichi Tanaka, who in the 1980s was working out the physics of responsive hydrogels. So these are molecules such as the polymer sodium polyacrylate. You add water, the water gets drawn in through osmotic force, you get expansion, and what happens afterwards is that it swells even more because the charged polymers repel, and you can get 1,000-fold or even more volumetric changes. The other thing, of course, is you have to get the polymers inside the piece of brain tissue if you want to swell it evenly. And to that, we owe a debt to some of these folks like Peter Hausen and Christine Dreyer at the Max Planck Institute, who around the same time, 1981, were figuring out how to take tissues and soak them in a solution of monomers to form polymer chains that form very similar hydrogels but uncharged. And they use that to facilitate imaging. You can make these samples much easier to handle and to image using microscopes. So let's put 2 and 2 together. If you could install polymer chains throughout in a very even way, but if you did it just right and you tuned the chemical composition to be hyperswellable, could you take a cell or a tissue and pull all the building blocks of life apart from each other until, like stars in a constellation, they are sort of hovering there in space, but the information is preserved. You're not going to lose the relative organization of the biomolecules that you care about. So to do that, we had to invent a couple chemistries in order to make this a viable idea. The first thing we did, as shown in this cartoon, was we developed anchors that can bind to biomolecules. So in this cartoon, the brown blobs are proteins, and we have developed anchors that will bind to proteins and equip them with little handles so we can apply force to them and pull them apart. And we've also developed handles that bind to DNA, to RNA, other kinds of biomolecules. And so now if you apply all of these at once, you can imagine someday an arena where you have biomolecules of all kinds being tied to convey force to them. Then you have to make the polymer. So we use a protocol that's very, very similar to the Hausen-Dreyer protocol from many decades ago, called free radical polymerization. You form these polymeric threads, and as they grow spontaneously, when they encounter one of the handles, they're going to bind to the handle, and the handle now becomes part of this network. And then finally, we need to loosen up the molecules from each other. They're loyal to each other, but we want them to be loyal to the polymer instead. So we can treat the specimen with detergents or heat or enzymes. And let's see. Whoops. All right, well, let's see if the movie will play. Ta-da! OK, with enzymes or heat or with detergents to loosen them up from each other or to destroy them if we don't care about them. And then when we add water, the polymers will swell just like Tanaka worked out many decades ago, but now the biomolecules will come along for the ride. So that result is something like the following. The polymer starts out compact, the brain starts out small, the polymer gets bigger, and the brain becomes 100 times bigger in volume, about 4.5 times bigger in each direction. So here's a little time-lapse movie of such a piece of brain tissue. This is from a piece of mouse brain. It's sped up by about 50-fold, so the whole process is more like an hour than a minute, but you can see it growing before your very eyes. And the polymer is so dense and so evenly made, the spacing between polymer threads is around 1 to 2 nanometers, we estimate, around the size of an individual biomolecule. So in various papers where we look at, and others have looked at, very small stereotyped things like microtubules or viruses, it seems to be even down to the very smallest dimensions that we care about in systems neuroscience. Across large scales, we can estimate the distortion. So you can do what's called a non-rigid registration, take a pre-expansion image and a post-expansion image, and then nudge them till they overlay. And the amount of distortion you get, you can then calculate. And so for all the protocols that we've published, we get distortions that are not zero, but they're pretty small, maybe a couple % over length scales of some tens or hundreds of microns. So what can you do with this? Well, there's a lot you can do with it. So one thing that you can do is to see the wiring of the brain much more finely. It's hard to see axons and dendrites and synapses because they are in the length scale of hundreds of nanometers, sometimes even a little bit smaller than that. Here is a piece of brain tissue that's so-called rainbow stained, where different epitopes are expressed in different cells, and then you bring in fluorescent markers that bind to the different epitopes. So every neuron gets a distinct color code. We didn't invent that. That was invented by Jeff Lichtman's group at Harvard. If you zoom in, it looks really fuzzy, though, because on a regular microscope, you just can't see very well. There's a limit in the resolution. You could do super resolution, but it would take forever to scan something this large. So if you expand, though, you can now see 1, 2, 3, 4 different individual projections. And so we and others are now working on a variety of algorithms to trace these neural circuits in a fashion that lets us reconstruct them. And not just reconstruct them, but to look at molecules in those cells, too. So one of the nice things about expansion is you decrowd all the molecules from each other. And so what you can do is to pull apart pieces of RNA, pieces of the genome, and so forth, and then we can run, we're collaborating with George Church's group at Harvard, sequencing on those very nucleotides and nucleic acids that are embedded within, in this case, a piece of mouse brain tissue. Or you can apply antibodies, one after the other, very quickly. And we're automating a lot of these processes. So the idea is that you could expand a brain circuit and map not just the wiring, but the molecules along those wires and at the connections. So that can be very powerful if we want to reconstruct brain circuitry with enough precision that you could, for example, simulate an entire brain computation in a computer. And one of my hopes is that for small brains, we might be able to do this kind of experiment in the coming few years. How big can you expand? Well, we came up with a kind of a fun little project. This is work that J.B. Chung did when he was in my group, where we polymerize a brain and expand it, like I showed you before. But then here's the cool part. You polymerize it again, and you can expand it again. So you get exponential spatial amplification with a linear number of steps. And so we call this iterative expansion. And so to make a long story short, you can get exquisite, nearly down to single molecule precision images by doing this. So here's a rainbow brain, color-coded like I showed you before. Expand it once, you can maybe map the pathways, but expand it twice, and you can see the very finest compartments of neurons, synapses and spines, and you name it. And if you look carefully, this image looks very speckly. It looks punctate, right? And that's because we really do think we're getting down to single epitopes, single proteins in these cells, which is something that we and others are now validating. Now, one of the fun things about neurotechnology is that the brain is such a high mountain to climb that if you solve a brain problem, you solve a lot of other problems as well. And so one thing we've been doing lately in work that was spearheaded by Yongshin Zhao and Octavia Bucur across my group and several pathology groups, Astrid Wein, Stu Schnitt, Andy Beck, is we've been expanding human cancer biopsies. So this is kind of a hard slide to see, but we've expanded a number of normal and cancer-containing human tissues. Maybe we'll go to the next slide because we can zoom in and see these things better. But to make a long story short, if you want to detect a disease early, you have to detect very subtle features, right? Because early means subtle, and subtle often means nanoscale. And so Stu had written an article a couple years ago about how early breast cancer lesions are very hard to diagnose. The disagreement rate between trained pathologists is something like 50%. So he insisted, and we agreed, let's see what we can do. So we expanded a bunch of conventional biopsies from human patients, and then we expanded the very same samples and imaged them again. And then we had a team of pathologists grade the diagnoses, and that became the fodder for training a machine learning algorithm to classify these different images. And if you did this whole assess and train ritual on the unexpanded images, this is the area under the curve of a ROC. So that's a fancy way of saying that 1.0 is perfect and 0.5 is chance in terms of the true positives and false positives. You want, of course, all the true positives you can get and as few of the false positives as you can as well. And pre-expansion, they were somewhere in between perfection and chance. But post-expansion, it was much better. And so this was a small-scale study. I wouldn't take the numerical values of these plot points too seriously. But we have actually spun out a company to try and do scaled-up clinical studies in order to see if we can revolutionize early detection of difficult-to-diagnose diseases. And it's taking off, so it's really easy to use. We teach people almost every week how to do this. We've trained about 300 groups in academia and industry just in the last year, frankly, year and a half. And people are applying it to all sorts of stuff. Can you detect bacteria, or look at human brain tissue, or map out peripheral nerves, or look at human parasites like Giardia? And the list goes on and on and on. So if you're interested in learning this technology, we have a very active teaching effort, and we run little mini classes at MIT nearly every week at this point. Okay, so we think we have a way now to map the wiring and molecules in static preserved brains, and we think we can apply it to a wide variety of problems beyond neuroscience. But what about the living brain? And so for the last part of the talk, I want to talk about two sets of technologies that allow you to read and write information to the living brain. So let's start with Optogenetics, which is a way to control high-speed dynamics of the living brain. And then we'll end on imaging, because this is sort of an extension of some of these earlier efforts. OK, so the idea here that my co-inventor, Carl Deisroth, and I came up with when we were actually both students around the year 2000 is, why can't we use light to control neurons? Because light can be aimed at things, and light, of course, is fastest anything ever gets. So if you want to really control the brain very precisely for prosthetic purposes, to remedy a disordered state, to input information in, light is not a bad way to go. And then you can bring light into the brain through any one of a number of device means that one can easily imagine. So the hard part, of course, which turns out to be kind of easy because nature solved it for us, is how to make brain cells sensitive to light, because the vast majority of brain cells don't respond to light. And so it turns out that in salty water, you find microbes that have membrane proteins that sense light and convert light into electrical signals across the membrane. And these were found half a century ago by people like Dieter Oesterheld and Walter Stolkinius, then in San Francisco, actually. And they found a 7-transmembrane protein that has a vitamin A relative that's a light-driven proton pump, which they named bacteriorhodopsin. And if you see in really salty water that has a weird color, there's a good chance you're seeing a microbe that has a pigment not unlike this one. About a decade later, a bunch of groups found light-driven chloride pumps, or halorhodopsins. And around the turn of the millennium, several groups found light-driven ion channels, which they called channelrhodopsins. And they all have relatively similar, although differing at key points, crystal structures. So what we've been doing is trying to figure out which of these will be safe and effective and fast enough to function in delicate, high-speed dynamic neurons. And so it turns out that members of all of these classes can be found that will work. And so we've been screening these now for quite some time. Light-driven proton pumps, for example, you can put into neurons. They'll pump protons out, and that'll hyperpolarize the cells. This was figured out by Brian Chow and Xue Han when they were postdocs working with me. And then chloride pumps will pump chloride in, and that was also worked out by Xue and also Amy Chuang when she was a student in the group. So these two methods let you shut down neural activity. And then light-driven ion channels, first myself collaborating with Carl Deseroth, and then later Nathan Klepecki when he was a grad student in my group, and others, we figured out how to use light-driven ion channels, the first of which we tried would bring positive charge into cells, thereby activating the cells. But in the time since, lots and lots of different molecules have been found with lots and lots of different properties. So where is the field headed now? Well, one effort is to make the technologies reach their physical limits of performance. Why can't you push them to make them sensitive to red light, for example? Because red light will go deeper in the brain or the body than other colors, right? This is a green laser pointer, and if I shine it through my finger, you can't see it. But I think we've all shined flashlights through our hands, and you can see the red glow or through your cheek. And that's because blood and tissue in general will absorb less red light than it will absorb other colors of visible light. So Amy, when she was in the group, worked out a way to silence neurons using a halorhodopsin that we called JAWS. And you could put the gene that encodes for this protein in with a virus, shine light, and then you could blockade neural activity very, very deep into tissue. And one of my colleagues, Bob Desimone, has since gone on to use this technology to inactivate tens of cubic millimeters in the monkey cortex, so pointing the way to potential uses in large brains. Nathan found this molecule crimson, which is a red light sensitive activator, just focused in the lower left. We're shining 735 nanometer lights. That's 100 nanometers redder than a red laser pointer. And we can then activate neurons very effectively, even though we're using infrared light. And actually, this molecule is entering human trials for blindness as we speak, actually, by a company in Paris. The other physical limit is space. So color's great. Red light will go deep in the body, but how can you be spatially precise? So one idea is, well, why can't you just holographically sculpt light and aim it at cells? And many people have been working on that over some time, including our collaborator, Valentina Emiliani. But there's a problem, namely that if you try to sculpt light to aim at a cell, you might hit the stray processes of nearby cells. And so for example, if you aim light at this cell, you're going to activate that one and that one also. So Urs Shemesh, Dimitri Tennesi, and Valeria Zampini, working across our groups, working on a collaboration to try to localize the light-driven ion channels just to the cell bodies of neurons. And then if you sculpt light to hit a neuron, you'll avoid the nearby neurons. So other people had tried this before. Maclean Bolton had pioneered one of the first such things, Xiaohua Pan, Frank Werblin. But they were all using the original channelrhodopsin that Carl and I put into neurons way back in 2004. And so what we decided to do was, well, why don't we try to screen all these novel opsins we're finding and see if we can make a superpower version. So we found a molecule, COCHR. We'd actually published it many years back, and we hadn't realized its use, or we would have come up with a cooler name. And so if you just express COCHR everywhere, of course, you get a lot of crosstalk. But to make a long story short, you can find a peptide and tack it on to COCHR, so it ends up just at the cell bodies. And now if you holographically sculpt light to aim just at the cells, you get effectively no crosstalk. So that's what we show here. Basically what we're doing here is we're looking at one cell, and we're trying to activate all of the other nearby cells. And the question is, how many times do you accidentally hit the cell that you're recording from inadvertently. And so if you use a regular molecule that's everywhere, it's like a third of the time. But if you stick the molecules just at the center of the cell, then it's effectively none of the time. So I think we're entering an interesting time now where maybe we could actually dial in activity patterns with single neuron precision and be able to control the cells throughout a circuit very, very precisely. So imagine, we're thinking of experiments that we could do, like could you enter in a specific pattern that could drive a decision or a memory in a very detailed way. And the applications of optogenetics are, into the many hundreds and thousands now, but hundreds and thousands now, not hundreds of thousands. But I thought I would just share one example because, akin to the theme of the conference, it's something that is going to lead to perhaps a wearable device, and also suggest a pipeline for discovery where we can take an invention, discover something about the brain, and then deploy a practical, wearable, non-invasive device into the world. This is work that was spearheaded by my close collaborator, ApplySci, and she and I have now co-founded a company to take this through clinical trials. Anyway, she used optogenetics to drive the brains of Alzheimer's model mice at a very specific frequency, 40 times a second. And that's sort of a magic number in neuroscience. But what she found was something that astonished even those of us working on the project, which is that if you drive the brain at that frequency, the amyloid plaques and the other molecular hallmarks of Alzheimer's, like phosphorylated tau, go down. So that's pretty shocking. Could you use a pattern of brain activity to clean up a disease which has a 99.6% failure rate for FDA approvals? But of course, if it's optogenetics, that's a gene therapy. People are not going to line up around the block for it. And so Emery Brown, another collaborator in the paper, who's an expert on brain rhythms and an anesthesiologist, suggested, why don't you try entering these rhythms in through our sensory pathways, through our eyes and through our ears? And so the team, which was spearheaded by Hannah Iacarino, who was a grad student with Leeway at the time, and Annabelle Singer, who was a postdoc in my group at the time, the team tried this. They tried having Alzheimer's model mice watch blinking lights on the ceiling. No optogenetics, no implanted fibers, no gene therapy, nothing. Just mice and an LED blinking 40 times a second. And amazingly, that was enough to also get rid of these problems of amyloid plaque formation and tauopathy and so forth. And so this led us to think, wow, movies to cure Alzheimer's. This would be very interesting if it worked. And many things, of course, fail to translate from mice to humans, but it's so simple to try that we thought we'd be idiots not to give it a go. So we've now started human trials, and hopefully we'll have useful news sometime this year. We're also working on non-invasive strategies for stimulating the human brain. We're lucky sometimes if we can enter information in through our senses. We're actually working on a huge variety of spin-off ideas related to this idea of using our eyes and ears as neural control pathways to help with disease. But some parts of the brain you just can't get to that way. And so when Nir Grossman was a postdoc in my group, we collaborated with several groups to try to figure out, could we exploit the nonlinear properties of the brain to aim for deep targets without hitting the overlying targets? And so the basic idea goes something like this. What if the brain had properties that are, to first order, like an AM radio? You would apply, so in an AM radio, you broadcast high frequencies through the air, and then the receiver will apply a nonlinear transform, could be as simple as a transistor for an old-fashioned transistor radio, and extract the low frequencies. And now you can hear somebody sing, or you can hear the news report. So the brain is also known to be nonlinear. So the question then arose, could you apply high frequencies, let's say in the 5 to 10 kilohertz range, to the surface of the brain, and then hopefully the brain would mostly ignore that. But where these frequencies overlap, maybe you could get interference that would result in low frequencies occurring at a point deep in the brain. So here's some data showing the idea kind of working, but for a surface structure. We played back 2 kilohertz and 2.01 kilohertz. And the neurons, that's what these dots are, the spiking of the neurons, they fire at the difference frequency, 10 hertz. If you apply just the high frequencies, you actually don't get a completely non-zero effect up front. The nonlinear properties of neurons will rectify, as it's known, these signals to some extent. But the effect is much, much smaller than what you get with the difference frequencies, even with the same amount of current. In fact, we're using a lot more current here than there, right? This is 100 microamperes. We're using 4 times higher current here just to try to be rigorous. And you do see something, but it's much less than that. So working with a number of people in Leeway's group again, we tried to figure out, could we activate the hippocampus without activating the overlying cortical structures? And to do that, her group used CFOS staining, which is a way of marking recently active neurons. It's a bit hard to see here, but there's a little green area here, and those are hippocampal neurons that are lighting up because we use this temporal interference method, and this is near the peak of where the activation should be. But we get very little activation overhead. Big bar in the hippocampus, very little in the cortex. If you apply high frequencies, we don't see much anywhere. And if you apply low frequencies everywhere, then you activate everything. So we're now working to improve the focality, to try this in humans, and also to figure out the path to, hopefully, medical testing and commercialization of this as well. Towards focality, one of the ideas that we're working on is a mathematical one. If you have two pairs of electrodes, like we did in the first study, you kind of get this football-shaped blob in the middle. So it's focal, but not super focal, right? But if you have many electrodes that sort of tile the space, maybe we could actually get extremely focal activation. And so that's something we're now working to test, working with Neer's group now at Imperial College London, and my group collaborating together. OK, so that wraps up control. I just wanted to end for the last couple of minutes on watching. So just today, actually, we published a paper on this. Watching is really hard. Maybe it's the hardest thing to do. And it's because you're dealing with a lot of constraints. You want to have a way to watch that's sensitive to small fluctuations. You want a way to watch that's fast. You want a way to watch that ideally gives you single-cell precision. And the list goes on and on and on. It's twice as hard as optogenetics, you could argue, because you've got to get the information in and back out, right? For optogenetics, you just send the information in. So in this paper we just published this afternoon, two postdocs, Erica Jung and Kirill Pyakovich in my group, decided to try to make a fluorescent voltage indicator. And this is a very difficult thing to do. So what did they do? Well, we decided that we needed to make a molecule that was bright, fast, stable, safe. And basically to do that, we had to do evolution, right? For optogenetics, we lucked out. Evolution did the evolution for us. But there's nothing in nature that broadcasts its thoughts through light. So we had to do the evolution ourselves. And so we built basically a robot that could do what scientists do in the laboratory. So we put mutant genes, it's called a library, into cells. And then we put those cells in a dish, and we scanned them. And if the gene product glows and is bright and stable and fast and so forth, then we bring in a robotic arm, and we pick out the cells that have all those properties. So we can screen for many different properties all at once. We only pick out the cells that are both bright and fast, and so on. And to make a long story short, we think we have now been able to filter through a huge number of these and come up with the first few prototypes that we think are extremely good in terms of their safety, stability, voltage sensitivity, brightness, and so forth. So these are just some early data. We actually have some in vivo data that I didn't have quite enough time to prepare for this talk, but this was done by Bernardo Sabatini's group in Brain Slice. And they can activate cells in one part of the mouse cortex and image cells in another part of the mouse cortex. And in black is what you get with an electrode, and magenta is what you get with the imaging. They overlay nearly perfectly. And down in the lower left are synapses. So you can see in black what the electrode records when you drive synaptic connections between the cells. And with magenta, you get the imaging. So it is not an ideal molecule. It's not as bright as we want. And so we're often using fairly bright laser powers when we are illuminating. But we've continued the screen. And my hope is that by the end of this year, maybe we'll have a molecule that not only is fast, well-localized, high dynamic range, and photostable like this one, but also is much brighter. So I think I'll end there. Obviously, we want to tie all these things together and to see if we can use these to derive the algorithms of how brains compute. And I think I've acknowledged all the people who've led the projects, but this is a massive collaborative effort. And so both our group members and alumni at the top and our close collaborators, which now looking at it's even longer than our group members and alumni, we're very indebted to the entire network of neuroscience for making all this happen. So thank you. I think we have some time for questions, if that's allowed.
3 minutes.
3 minutes. Yes?
So transcutaneous electrical stimulation, widely used in medicine, PTA for pain, so on. Some people use a modality called interferential current. Any thoughts about whether that mechanism might account for the efficacy and safety as you study that molecular or at the cellular level?
Yeah, great question. So we were trying to use levels of current that were much lower than those because we wanted to avoid stimulating peripheral nerves in the scalp. So my gut feeling, although we haven't studied peripheral nerves, so take this with a grain of salt, is that maybe these mechanisms could be different. But right now, one of our main foci in our group is actually to understand the mechanism. So we're doing a lot of electrophysiology, and maybe we'll do some imaging as well to try to probe the mechanism with much finer scale accuracy. So the honest answer is we're not sure yet, but hopefully we'll know sometime in the coming months. Other questions? Yes?
Have you done any work with Optogenetics in your lab to sort of combine up like the genetic component with safe levels ultrasound or something like that?
We have not. No, we've been very focused on trying to get down to single cells and more fundamental building blocks of neural circuits. But it's a very exciting area, and there's also magnetogenetics and all sorts of renaissances going on in different intersections of genetic tools and physical tools. Okay, well, thanks.